All right, so, sorry, my camera died on me and I had to switch batteries. Okay, so I might not edit this and I might just make it a part 2 because beggars can't be choosers and I'm not an editor, so I don't know how to make stitch videos together. If somebody knows how to do it, they can help me out. So I left off, I believe, at the point where we were talking about how the, basically like the myosin heads bound to the exposed active sites after calcium had pulled, bound to troponin and pulled tropomyosin off of the binding sites. Yep, sound right? Hopefully. Okay, so what's going to happen is a molecule of ATP is going to be attached to this head, this tropo or this myosin head on the myosin thick filament. That is going to allow it to be moved into a position or cocked up into a position where it can be holding on or attached to the actin binding sites. Alright, so that's step 1. Now when that happens, when we get that binding, we phosphorylate it and we get basically like a ADP and a phosphate molecule. Okay, so it's bound and it's in like a state like this. Here's our actin, and here is our myosin head coming off of myosin. We've got our ADP and our phosphate group, which are both still bound to this myosin head. Okay, now that's all good and well. We kind of did 2 good steps in making a muscle contract. We've got it bound and it's ready to the point where it can actually move. Okay, so what each of these individual fibers on myosin will do is they are going to go ahead and push the phosphate away. This is like the rocket fuel. Okay, so when it gets rid of this phosphate group, you're going to make this head kind of do like a bicep curl. So what's going to happen is it's going to go like this. Now it's going to be down here. So it's going to be shifting the actin filament that way. Okay? So we went from here to here, and it's pulling. I'm not the best at drawing, so you can imagine, but it's pulling the actin filament towards the midline, the M line, okay? And since that actin is bound to the Z line, it's going to be pulling the Z line with it towards the M line, okay? The myosin never really moves, but the individual heads on it do move. All right? And now the final step to contraction, which is why I recognize, or I led with rigor mortis, is we've got a state where the myosin head is doing a bicep curl and pulling these towards the center, pulling the Z lines together. Okay? And we need to unbind it so the muscle can relax, or this process can start over again, because we have a lot of space between the 2 actin filaments of each Z line. And if we keep going with this process and rebinding ATP and making those power strokes and releasing that phosphate group, we're going to be able to basically crawl these Z lines almost to the point where the actin touches and shorten it. And shortening, we all know, is how muscles contract. So we need to get rid of this. So in order for a myosin head to unattach from actin, we have to have fresh ATP come in and bind to the head. So when ATP comes in and binds the head, it can now go ahead and return back to its resting position. Okay? And now all these different heads here are going to be in different points in the cycle. So they could have a head that's bound over here and a head that just finished contracting and got unbound over here. So that way, when you're trying to create a synergistic effect and shorten the two Z lines, every time you release the myosin head from the actin, the whole thing doesn't just recoil back out because you've still got some heads down. Okay? So that is basically that. And what happens with rigor mortis? Why did I leave with it? I keep saying it and I forget to actually tell you about it. Rigor mortis is when a person dies, they don't make ATP anymore. So for a little while, there's ATP, a few seconds of ATP stored up, and then you can undergo anaerobic respiration. You'll still have some oxygen in your blood, so you make ATP. But after a while, your ATP runs out. And when it runs out, your myosin heads can't unbind from the tropin, or from the actin binding site, Tropin binds calcium. And when that happens, your muscles are stuck in a contracted state, and that leads to rigor mortis, which happens 4 hours after death. So yeah, that is skeletal muscle physiology. Let me think if I'm missing anything here. Let me look at the notes. The only thing I forgot to mention was, what is the triad? The triad is a T-tubule with a sarcoplasmic reticulum on each side. So there's going to be 2 of them. Here's an SR, here's an SR. It's going to be on both sides so that depolarization can release lots and lots of calcium, so that we can go ahead and bind the calcium to troponin, pull it off of the actin, or it can pull off tropomyosin from the actin binding sites, and the myosin heads can bind to the actin, and when they get ATP, they can bind to actin, and then they can initiate a power stroke, getting rid of a phosphorus group, and then when they need to unbind, ATP comes in and binds again, allows them to uncock, alright, and then we can start the whole process over again. So it's just a big cycle. There was 2 disorders he talked about with this. So we have these integrin molecules or proteins that span the plasma membrane and out to the basal lamina. And then we also have the dystrophin protein. So with dystrophin, we have this is the dystrophin, this is the actual dystrophin molecule. That is going to be bound to a specific part on each sarcomere, okay, in the cell. And it's going to function to kind of attach it to the cell wall, okay? So we actually get some shortening of the entire cell and not just the myofibrils in it, okay? And I forget which part it binds to. It's going to bind to actin. So this dystrophin molecule right here that comes, this is the plasma membrane, is going to bind right up here to one of the points of actin near the Z line. So that's going to tell the cell membrane to fold up too as the sarcomere contracts. That's skeletal muscle physiology.